Microfluidic platforms for personalized medicine and pharmacogenomics are transforming healthcare by making treatments more precise and tailored to each individual. Have you ever wondered why the same medicine can work well for one person but cause serious side effects for another? This is because everyone's body processes drugs differently due to their unique genetic makeup. Microfluidic technology is helping solve this problem by allowing doctors and scientists to quickly analyze a patient's genes, proteins, and metabolism using just a tiny drop of blood or saliva. Instead of relying on a one-size-fits-all approach to medication, we can now develop treatments that are perfectly matched to a person's biology. For example, in cancer treatment, doctors often need to decide which drug will be most effective for a particular patient. But what if we could test multiple drugs on a small sample of a patient's tumor cells before making that decision? Microfluidic platforms make this possible, helping doctors select the most effective and least toxic treatment for each individual. Similarly, in pharmacogenomics, the study of how genes affect drug responses, microfluidic devices can quickly detect genetic variations that determine how someone metabolizes medications. Imagine someone taking a common painkiller. For one person, it may work perfectly, but for another, it could be ineffective or even dangerous. With microfluidics, doctors can determine the right drug and the right dose for each patient, reducing trial and error and improving safety. But the applications go beyond just medicine. Think about infectious diseases. How do doctors know which antibiotic will work best for a bacterial infection? Microfluidic platforms can rapidly test bacteria from a patient's sample against different antibiotics, helping doctors choose the most effective treatment immediately rather than waiting days for traditional lab results. This could be life-saving, especially in critical infections where time is of the essence. What if this technology became available for home use? Imagine a small, portable device that could analyze your genetic makeup and tell you which medications work best for you before you even take them. Could this change how we approach healthcare entirely? Instead of guessing or waiting for symptoms to appear, personalized medicine could allow for preventive care and early interventions, improving health outcomes and even saving lives. The future of medicine is shifting towards precision, and microfluidic platforms are playing a key role in making that happen. By offering fast, cost-efficient, and highly accurate analysis, they are bridging the gap between cutting-edge research and real-world healthcare applications. Whether it's cancer treatment, drug safety, or infectious disease management, microfluidics is paving the way for a future where medicine is not just about treating diseases, but about treating people as individuals. How might this change the way we think about healthcare in the next decade? Could microfluidics bring truly personalized medicine? To every home and hospital, the possibilities are both exciting and limitless. Microfluidic platforms allow precise control over fluid flow, enabling high-throughput drug screening on patient DNA cells. Traditional drug testing relies on large-scale experiments that often fail to account for individual genetic variations. Microfluidic devices, on the other hand, can replicate physiological conditions at the micro scale, providing a costly and rapid means to evaluate drug responses in personalized medicine. This technology facilitates the development of tailored therapies by testing drug efficacy and toxicity directly on patient samples, reducing the likelihood of adverse effects. A microfluidic-based organ-on-a-chip model replicating liver metabolism has been used to predict individual responses to chemotherapy drugs, minimizing trial and error in cancer treatment. Microfluidic drug testing works like a custom-tailored suit designed to fit an individual's unique biology rather than using a one-size-fits-all approach. Pharmacogenomics studies how genetic variations affect drug metabolism and response. Microfluidic systems enable rapid genetic screening by processing DNA and RNA from small biological samples. By integrating microfluidics with next-generation sequencing, NGS, and CRISPR-based diagnostics, Researchers can identify genetic markers that influence drug absorption, distribution, metabolism, and excretion. This allows for personalized dosing strategies, ensuring maximum therapeutic benefits while minimizing side effects. Microfluidic DNA chips are used to detect genetic polymorphisms in cytochrome P450 enzymes, which influence how individuals metabolize antidepressants and cardiovascular drugs. Pharmacogenomics with microfluidics functions like a genetic roadmap, guiding precise drug choices based on an individual's biological makeup. Lab-on-a-Chip technology miniaturizes laboratory functions onto a single device, enabling real-time monitoring of drug levels in the bloodstream. This is particularly crucial for patients taking medications with narrow therapeutic windows, such as anticoagulants and immunosuppressants. By continuously measuring drug concentration, microfluidic LOC devices help adjust dosages in real time, preventing underdosing or overdosing, thereby optimizing treatment efficacy. A microfluidic device developed for monitoring warfarin levels in blood has enabled personalized anticoagulation therapy, reducing the risk of stroke and bleeding complications. LOC devices work like a smart thermostat, constantly adjusting and optimizing drug levels to maintain balance in the body. Cancer treatment is shifting toward precision oncology, where therapies are tailored to the genetic and molecular profile of a tumor. Microfluidic platforms facilitate ex vivo tumor modeling by culturing patient tumor cells in controlled environments, allowing oncologists to test different drugs before administering them. This personalized approach improves treatment outcomes by selecting the most effective therapy while avoiding drugs with poor efficacy or toxic side effects. A microfluidic tumor-on-a-chip model has been used to test the response of breast cancer cells to targeted inhibitors, helping oncologists refine chemotherapy regimens for individual patients. Tumor-on-a-chip technology is like a simulation test for cancer drugs, identifying the best treatment before exposing patients to unnecessary side effects. Neurodegenerative and psychiatric disorders exhibit significant variability in drug response among individuals. Microfluidic-based neural models, such as brain-on-a-chip systems, replicate the microenvironment of the nervous system and allow personalized screening of neuroactive compounds. This technology is particularly valuable for conditions like Parkinson's and epilepsy, where individual responses to medication can vary widely. Microfluidic platforms have been used to test the effects of dopamine-based treatments on patient-degree neurons, enabling tailored Parkinson's disease therapy. These devices function like a personalized tuning system, fine-tuning medication to match the unique neurological profile of each patient. Biomarkers provide critical insights into disease progression and treatment response. Microfluidic platforms integrate biosensors capable of detecting molecular biomarkers in blood, saliva, or urine in real time. By continuously monitoring biomarker fluctuations, microfluidic devices support dynamic treatment adjustments in chronic conditions like diabetes, cardiovascular diseases, and autoimmune disorders. A microfluidic biosensor for real-time glucose monitoring has improved personalized insulin therapy for diabetic patients, reducing the risks of hypo or hyperglycemia. This system works like a real-time GPS for health, constantly updating treatment routes based on biomarker feedback. Antibiotic resistance is a growing global concern. Microfluidic platforms enable rapid bacterial culture and susceptibility testing, identifying the most effective antibiotic for each patient. Traditional antibiotic selection methods take days, but microfluidic assays accelerate the process, reducing inappropriate antibiotic use and preventing the development of resistant bacterial strains. A microfluidic-based bacterial culture system has successfully determined antibiotic resistance patterns in urinary tract infections within hours, improving targeted treatment strategies. This approach functions like a detective solving a medical mystery, quickly identifying the right antibiotic to eliminate bacterial threats. Microfluidic-based drug carriers, such as liposomes and nanoparticles, enable site-specific drug delivery, reducing systemic side effects. By mimicking physiological conditions, microfluidic synthesis techniques optimize drug encapsulation and release kinetics, ensuring that medications reach the intended organ or tissue with maximum efficacy. Microfluidic generated lipid nanoparticles have been used for targeted chemotherapy delivery, reducing damage to healthy cells while increasing tumor penetration. Targeted drug delivery works like a guided missile, precisely delivering medication to the disease site without collateral damage. The future of personalized medicine lies in the convergence of artificial intelligence, AI, and microfluidics. AI-powered microfluidic platforms analyze vast data sets from genetic, proteomic, and metabolic profiling to recommend optimal treatments in real time. These intelligent systems will transform personalized healthcare by automating diagnostics, predicting disease progression, and tailoring therapeutic regimens with unprecedented precision. AI-integrated microfluidic labs are being developed to process patient samples and instantly generate personalized treatment recommendations for complex diseases like cancer and autoimmune disorders. This system operates like a self-driving car for healthcare, navigating complex biological data to deliver the most precise and personalized treatment possible.